All right, folks, I'm going to do um, probably the last lecture on viruses, and then I'll be structuring an exam, and we'll be posting that by week's end, right? So that we can get that in. We need to do 5 exams. Again, I'll give you an extended period of time to do it. Um, doing the best we can. So, um, anyway, let's look at this last segment of this particular PowerPoint, this lecture. So after viral infection, as I said, viruses either go into the cytoplasm, their genome goes there and is operative. You have assembly of viral parts, and viruses start coming together there. Or it can go to the nucleus. In the case of DNA viruses, they go to the nucleus and they exhibit their effects there. And so infected cells, the upshot is infected cells will exhibit evidence of viral infection. And sometimes the type of effects we see are so specific that it lets us identify the particular virus that's causing the infection. And so if you were a pathologist and you were charged with, say, diagnosing a viral illness, you might look for these so-called cytopathic effects. Right? Cytopathic effects. Cyto means cell. Pathic refers to pathology, disease states. And so cytopathic effects are visual evidences of viral infection. So what are the kinds of things that we might look for? Well, one would be changes in size and shape. Frequently, virally infected cells will start to get a little bigger, a little fatter, and they will, if they have a specific shape, say they're cuboidal to start. The cell in its normal state is cuboidal, like in the renal tubules. If it's virally infected, it'll lose the cuboidal shape and it'll round out, and so it'll look swollen and just take on this sort of very generalized look. And the reason for that has to do with the loss of protein. So viruses are using protein like crazy to build their capsomers, and so the host cell has its proteins basically dismantled so the virus can use them. And one of the main sources of protein for the virus would be the cytoskeleton. Remember, cells have cytoskeletons that kind of help them retain their shape, maintain their shape. So loss of the cytoskeleton may lead to this rounding out. And of course, other proteins that are soluble in the cytoplasm, they're lost too. And the cell may then lose its capacity to water balance and that sort of thing. That could cause this rounding out as well. So we could look for that. In other cases, cells actually may shrink a little bit. And it's loss of parts that are contributing to that sort of thing. If, say, the cell is budding lots of viruses and the membrane is going away, it's going to get smaller and smaller. That may be a consequence too. Cytoplasmic inclusion bodies. So those could be vesicles containing viruses that have been taken up through endocytosis. And once they enter, if that vesicle envelope combination is not disrupted or not destroyed but remains behind, if the genetic material is simply extruded through that, you can see these little granules start to show up. So cytoplasm that normally would be not so granular might start to look more granular as viral infection proceeds. That's a possibility. There could be, inclusion bodies, say in the nucleus, so or in other compartments of the cell. Those would just be granules again. Number 4 is pretty interesting. So multinucleated cells. Um, this happens when the the boundaries between cells that keep them discrete are lost. Okay, so if you've got let's say here's a patch of cells, all right, a group of some cells here. I'll put 4 of them or so. Horrible looking cells, right? But they're discrete and they're separate and they're functioning independently of one another. These little squigglies inside of here are supposed to be their nuclei. Can't do that so well with my finger. But what keeps these cells separate in part would be proteins that are embedded in the membranes. All right, there's these little proteins that help to bind the cells and allow them to communicate without actually fusing together. And so you've got gap junctions and tight junctions, you may recall that sort of thing. Well, again, these are composed of protein. And what do viruses do? Well, they use up protein. And so if the proteins are lost, right, if the virus starts eating up these proteins, you know, the boundaries between the cells are lost, and they'll eventually all sort of meld together. Here then is all of those cells, however many I had before. I think there were 5 of them up here. They all just sort of fuse together. And it is going to take on the appearance of one big cell with several nuclei, right? So giant cells with many nuclei, that's called a syncytium. SYNCYT. IUM, syncytium, syn together, CYT refers to cells, bunches of cells fused. And so that would be an indication of viral infection. You start to see that. That's not a normal thing. Or cell lysis, viruses accumulating in a host cell and then eventually exceeding the limit that cell can hold, and it bursts. And so if you see tissues where the cells usually are very tightly packed, starting to exhibit lots of cell loss and disruption of the completeness of that tissue, well then that could be indicative of a viral infection. DNA viruses go to the nucleus. And even some RNA viruses, the retroviruses, reverse transcription happens and DNA is produced using an RNA template. Well, that DNA goes to the nucleus as well. And so DNA is getting incorporated into chromosomes of host cells will change the shape of the chromosome. And if you were to look at those cells while they're in the process of cell division, you'll see the chromosomes taking on unusual appearances. They'll get longer than they should be and different things like that. So you'll see evidence of infection there in the nucleus as the DNA is altered. And then this last step is related to number 6. Number 7 is related to DNA alteration, because if you have a piece of DNA that gets plugged into a functional genome, depending on where the virus DNA is embedded in the genome, it can affect how that host cell works. It can turn on or turn off certain genes in the host cell. If, say, one of the genes that gets turned off is a gene that codes for something that controls the cell cycle, the host cell will lose its ability to regulate the cell cycle. Okay? Which means that the cells, instead of having this nice regulated sequence of being active and then dividing, so if division is is not controlled, the cells divide out of control. They multiply in number way beyond what they're supposed to. And what do? You get a tumor, right? So tumor formation can happen when there's this transformation. We call it transformation. When viral DNA gets incorporated into a host cell's DNA, it can transform the cell, right? And if tumor forms, and that metastasizes, obviously that's cancer. And there are a number of viruses that can do this. And there are some people who are seriously considering the possibility that all cancers have a viral origin somewhere along the way. That there's research looking into that. But we know for sure that human papillomaviruses, those that cause warts, which there's a bunch of different ones of those are DNA viruses. And then the Epstein-Barr virus is another one. Those are well known as triggers for tumor formation and ultimately cancers. So viruses can do that. So we can look for evidence of cancerous cells. That could be an indicator of viral infection. So those are all cytopathic effects. Then we come to the topic of persistent infections, right? So I'm going to go ahead and make a change here right now because I don't like how they've worded this, and I always forget to take this out. But there's this, I can't erase that. But this term, let's do this. Let me take out the term chronic for now. And we'll talk about a class of viruses that cause what are called latent infections. There's different kinds of persistent infections. So persistent infection is where you have a viral infection, right? Host cells are infected, and it's a prolonged infection. It takes a long time for it to be carried out. All right, and so one type of persistent infection that you can sometimes have, right, is a latent infection, all right? So latency looks like this. Let me draw a little graph that illustrates this. So here's a graph, and on this axis down here is viral load, which, that's the number of viral particles that would be evident in cells, right? And then on this axis, the y-axis would be, the appearance of symptoms. I don't know. So initially, there are no symptoms. And then with these latent persistent infections, what happens is they build. And then you become symptomatic, right around in here. The viral load increases. We could also put time on this axis too. That's the same. You become symptomatic right about there, right? Takes a while. And the key is, let me change that. You become symptomatic right about here, and then you exhibit fewer signs. Now the signs start to diminish, and you go back to a condition where you're no longer symptomatic. Now you still harbor the virus. And so that's a state of dormancy. So that is typical of a latent type of persistent infection. Now, sometime later, you can have a trigger that causes it to become symptomatic again. And this could happen repeatedly throughout your lifetime. So look at the viruses that are typically like that. Measles virus, if you were to, God forbid, be infected by measles and you don't clear that entirely, you could develop symptoms of the measles infection for years and years afterwards, right? The herpes viruses do this same sort of thing. So we have these flares with herpes infections that can sometimes happen, especially with the genital herpes. Herpes zoster, or I think they're calling it varicella now, that's chickenpox. And, you can have that as a child, but then later you still harbor that virus, you can develop shingles, which is a much more serious condition. All right, so there's that. Actually, on this axis down here, I apologize, I messed this axis up. This axis down here is time. I'll get it right. That's time. And then on this axis here, this is viral load. All right. That's just simply the number of viral particles that your cells contain. And so over time, you can see this viral load goes up and down. And in these low spots in here, these valleys between the peaks, you're in dormancy. Okay? That's latent. That's the latent type of infection. They're persistent, they're long-lasting, and they're latent. They display these periodic, spikes. All right, so let me throw in here. Let me stop this. I'm going to insert another slide because I want to show you two other variations of persistent infections. So another one is the chronic type of infection. Let me write that in here. Chronic infection. This is a type of persistent infection as well. A second type of persistent infection, one that's long lasting, all right? And so this is where you would have, if I could use my, here we go. This is where you'd have a graph that looks like this. Chronic, time on this axis. There's our viral load over here. Number of viral particles that you're carrying. Initially, you don't have much, but then their numbers build. And this is an exponential increase in the number of viral particles that you're carrying. See, it's got this little bit of a J shape to it. But at some point, it maxes out, and you've got a whole lot of virus. And it stays pretty much at that level. And it's at this point where you develop the full slate of symptoms that are typical of whatever viral infectious disease that you have. So there are some viruses that do this kind of thing. And this goes on, and you display those symptoms throughout the entire period that you're infected, as long as you're still alive. It may terminate with your death in some cases if it's really extreme, but it never goes back down here to dormancy. That doesn't happen. It just stays pegged out at that maximum level. And that's a chronic infection, very long lasting, right? And so the hepatitis viral infections, in particular Hep B, is like this. Okay, it's a chronic type of infection. So that's a second type of persistent infection. Some others, I'm sure, that look like that. Then there's a third one, right? Let me insert another slide real quick. Slide. We do this one. So another type of infection is what we call a slow infection. This is persistent too. Third type. Third type of persistent infection. All right, so that would look like this. This is where you've got your graph, viral load over here, right, time here. And often these types of viruses cause a really acute, abrupt appearance of symptoms. Results in that you're symptomatic here, and then it declines, and you don't display much, if anything. You might appear to be totally asymptomatic when it drops back down here, but you still carry the virus. And for a really long time thereafter, I'm telling you what, or maybe years thereafter, builds until finally the full slate of symptoms happen. That's called a slow infection. And like I say, it can take decades for it to play out. And of course, once it gets up into here, your body's so weakened at that point that boom, usually, mortality, fatality is the end point of that. But this is what we would expect to see with an HIV infection. It's a slow infection, a third type of persistent infection. So you've got those that look like an S-shaped curve. I lost that. An S-shaped curve. That would be typical of a chronic type of persistent infection. And then you have the other one where you have latency, as is the case with herpes virus, the measles, where it can spike and then go back down to dormancy and then spike and go back down to dormancy. So those are different variations of those. All right, so we have those. And then you have infections like you would say get if you had a rhinovirus, a cold virus infection, and it would look like this. Get the virus, you develop symptoms, and then it goes. So you can clear viruses. That would be an example of an acute type of infection that's not persistent. We have that. A lot of viruses are persistent. They just sort of hang around for long periods of time, and frequently they are of the latent type, where you get acute sort of appearance of symptoms, and then it drops back to dormancy, and you just stay dormant for the rest of your life without any blips on the screen. You may still carry that virus. All right, so there's that with regard to animal viruses. Multiplication cycle, the different types of infections that you can have, persistent forms that are chronic or latent or slow. And then we have the bacteriophages, or phages. I don't know. I've always pronounced it that way. I had a professor who pronounced it like that, and I just mimicked him, and I continue to pronounce it that way. But these are viruses that infect bacteria. A lot of people, I think, don't think that can happen, but it does. In fact, there are viruses that infect amoebas, viruses that infect all of the different kinds of fungi, viruses that infect all of the different kinds of arthropods. So there's grasshopper viruses, and there's crayfish viruses, and starfish viruses, and on and on and on. If you're a living organism, you're going to have viruses. That's just simple as that. All right. So for every living thing, there's probably several, who knows how many, dozen or hundreds or thousands of viruses that you can get. So it's just mind-boggling the number of type of viruses that could potentially be out there. So the bacteria are infected by their own types of viruses that are unique to them. We call them phages, because the root phage refers to eat. So if you have, say, a colony of bacteria growing on an agar plate, they become infected with one of these viruses, they'll literally just wipe that colony out or that film of growth out, and you won't see them anymore. That's how destructive they can be to the viruses or to the bacteria. There's some very commonly studied ones of these. One very famous one is called T4. The T4 phage is one that infects E. coli sort of preferentially. And so they've used it to introduce genetic material into an E. coli to transform it and do different things. T4 was also really important in trying to figure out exactly how the genomes of organisms work. So the DNA that's contained in that virus is what's introduced into the host cell, and what happens thereafter be indicative of how the genetic. But anyway, so T4 was heavily studied back in the 20th century and continues to be an important virus in molecular and other kinds of studies, genetic studies. These are complex viruses. As we pointed out, they've got the icosahedral capsid, but they have the other parts, the tail fibers and the sheaths, all of that. And they are principally DNA viruses. They can be double-stranded or single-stranded. There are a few RNA forms among the bacteriophages, but not many. The cycle that they go through is similar to that which we see with animal viruses, but there's some subtle differences. We'll talk about that. Well, we can talk about it right now because out here, uncoding is not something that has to happen because the capsid never goes in to the host cell. It remains on the outside. The genetic material comes in free of any sort of capsid or envelope or anything like that. And so that step is cut out. That's the main difference between what happens in bacteriophages and the animal viruses. Now, thereafter, though, once that genetic material is in the host cell, there's different directions that can be taken. Right. So frequently, what we see is a lytic cycle that's carried out, and this is where the genetic material, the DNA, codes for messenger RNA. The messenger RNA codes for protein. Capsid parts are produced. New genetic material is produced. Viruses are assembled, and boom, the host cell goes through lysis. That happens abruptly. Bacterial cells die as a result of that, and that infection goes, from beginning to end very quickly. All right. That's one possibility, and that's probably the most frequent possibility. But as we'll see, there's another type of endpoint that can be taken that differs from that. Okay? So bear that in mind. But here are the steps in replication of the bacteriophages. You've got adsorption. You're going to have the penetration of the genome, and that's all that enters. Thereafter, there is no uncoding. So between 2 and 3 here, we don't have an uncoding step. Replication or synthesis is something that can happen essentially immediately. And then assembly, maturation, and lysis would be commonly what we would see. Now, as I said, with some viruses, instead of going right into lysis, they may go into another sequence of events. And so that's what's shown here. All right, let's work through this. So here's the beginning. The host cell is this guy right here. It's like an E. coli, a rod, bacillus. Here's one phage. Now, typically, when these guys infect, they absorb in large numbers. You have dozens of them, maybe. But for purpose of explanation, they just show the wand. And you can see that the tail fibers have attached. It plunges this sheath with the little pins at the base of it down, and it penetrates the cell wall, cell membrane, the envelope. And here's our genetic material, the bacterial, the viral DNA coming in. Here's the bacterial genome, the nucleoid. There's penetration. And immediately that DNA can be transcribed into messenger RNA, which then can be translated. And here you have in this step, you can see evidence in that cytoplasm of all kinds of particles coming in. Viral capsid parts and genetic material. And so all of that is being produced. And here's the assembly step, and you can see that more, virus-looking things are being produced. Capsids are coming together, sheaths are coming together, tail fibers are coming together. And then you can see there's maturation. These are full-fledged viruses. At this point, because they're still inside of the host cell, they're called virions. But if they could escape, they would be able to, be competent to infect new host cells. And that, of course, is what happens here. This is lysis. And so there's the rupture of the host cell, and boom, and you got new viruses escaping, and they're going to go find new hosts. Now that's the Lytic cycle. That's sort of the standard. That's usually what happens. As I said, occasionally viruses don't do that. They infect. There's penetration, but then they slip immediately into this step. So you can see here what we've got in the nucleoid. I can take that nucleoid out. The nucleoid of the bacterium has incorporated into it a piece of viral DNA. So the blue in there is viral DNA plugged into the nucleoid, all right? It's a recombinant DNA. Remember that? We talked about that previously. This is a recombinant DNA. And so that is not going to produce symptoms, that are overt. The bacterium can persist. It can live. In fact, this may be advantageous to the bacterium. Bacterium is not killed outright. It may actually function differently and even better than it did before with the virus incorporated into it. That's just really bizarre that happens. But that's called lysogeny. All right, this is the lysogenic state. And what will happen is that virus can go through binary fission. It'll copy its genetic material, and it's going to copy the viral DNA right along with it. So if that one virus divides to form 2, and those 2 divide to form 4, and those 4 divide to form 8, it goes on and on like that. The number of viral DNAs that are going to be present is going to be exactly the same as the number of bacteria that are present, right? And that'll go on for however long. In fact, it may just continue permanently that way. But it doesn't necessarily have to happen. Sometimes the virus will come back out of of the lysogenic state and go right back into the lytic cycle. So there may be some trigger for that, maybe a stressor on the bacterial population. They run out of food, they run out of water, temperature changes that may trigger this sort of reversal, going from lysogeny back into the lytic cycle. But yeah, so when the phage is incorporated into the bacterial nucleoid in lysogeny, we refer to that as a prophage. Okay, so this is the prophage here. Is the recombinant nucleoid combined with the viral DNA. It's a prophage. It's essentially a virus laying there dormant. That's what a prophage is. You see that in lysogeny. Right? And like I said, it can be advantageous for the bacterium if it's infected this way. That's interesting. All right, so we'll come back to that. I think there's another slide later that revisits that and some of the consequences of it. So you've got the lytic cycle and lysogeny. Lytic cycle is typical. Lysogeny is something that is sort of special, and only certain viruses do that. All right, and when lysogeny happens, the viral DNA incorporates into the nucleoid, and we've got a prophage. Okay, and that can be reversed. You can go back out of that back into the lytic cycle if conditions warrant that later. So which of the following is a step found in animal virus multiplication but not in bacteriophage replication? It should be letter C, right? So it is in fact letter C. That has to happen in animal viruses. And the bacteriophages, it's not something we would expect because the capsid doesn't even come in with the phage. Yeah, so here's lysogeny again. And so we can think of this as it says here, a silent type of virus infection of bacteriophages. So yeah, the forms that do this, that carry out lysogeny, are known as the temperate phages. Temperate means they're even tempered. They're not really destructive usually. Kill outright. So if you're a temperate person, you're a kind and equable person that doesn't have a temper that flashes and goes up and down. That's kind of what they're trying to imply here with that. Then, like I said, the prophage is the viral DNA plugged into the bacterial DNA. That's what we call a prophage. Induction is the name of the phenomenon where the prophage comes back out of dormancy and becomes active again. So this is activation of the virus. Then it goes into the lytic cycle. So this is going into cell lysis as induction. Those are some things that would apply here when we're talking about these lysogenic forms, these temperate phages. All right, so like I said, sometimes lysogeny can be advantageous to a host cell. Doesn't kill the host cell, but provides it with advantages. And this is unfortunate because the advantage for the bacterium is that it becomes pathogenic as opposed to just being, say, a commensal or being free-living. It becomes pathogenic. It can cause infection and disease in human and other hosts. And so this is called lysogenic conversion. This is where a bacterium is infected with a virus. It's a temperate phage. There's this formation of the prophage, the combination of nucleoid and viral DNA. By virtue of where the viral DNA inserts into the bacterial chromosome, it can alter how that bacterium functions and cause that bacterium to start producing toxins and or enzymes that can cause disease, pathology. And so some of the bacteria that are affected in this fashion are Corynebacterium diphtheriae, This is the causative agent of diphtheria, this horrible respiratory illness. Now, if C. diphtheriae, Corynebacterium diphtheriae, is not virally infected, it's not a pathogen. It doesn't cause disease. So it's only when it's lysogenically converted that that'll happen. Same with Vibrio cholerae, and this causes cholera. Remember, it's a Vibrio, so that means it's got this sort of comma-shaped morphology. It's a rod, but it's bent into a C, so that's typical of the Vibrios. But these live in usually brackish water or sort of mildly salty water along coastlines. And so if you were to, say, wade out into that water, or God forbid, get some of it in your mouth or in the mucous membranes, and if this particular bacterium was lysogenically converted by a virus, it could cause cholera and lead to death fairly quickly. And then botulism, food poisoning, is caused by a Clostridium species. And remember the genus Clostridium? Those are gram-positive rods that are spore formers. These are endospore formers. And this particular species, botulinum, is the causative agent of botulism, food poisoning. So if you don't can your vegetables properly, if you don't use a pressure canner to sterilize and kill all of the microbes that are on the food, you could wind up with botulism if you were to eat those, say, green beans, that were not properly canned, because the Clostridium botulinum continued to persist in there. And the reason it produces the toxin, the botulism toxin, is it's been lysogenically converted by a virus. So there's a number of forms that do that. They're infected by a virus, and that's what makes them bad. If they're not infected by the virus, then they're okay. They're not problematic. They're not pathogenic. All right, so the last thing I want to talk about has to do with how we study viruses. How do we know what we know about them? A lot of the same things that are done with bacteria, culturing them on different kinds of media and acquiring enough of them to be able to study them under the microscope and other ways, a lot of that stuff can be done with viruses, but we can't use the same media. We can't use the same approaches because, as it says here, viruses are obligate intracellular parasites, which means we can't just place them on a nutrient agar or, in a nutrient broth because there's nothing in there for them to infect. So they have to infect something. They have to parasitize something. So the cultures that we come up with involve utilizing actual live cells. And so what was developed back in the 20th century, about midway through, was this technique of cell culture where we take stem cells, which are generalized cells that don't have a definite function yet. They can come out of bone marrow or stem cells or other sources. You have stem cells in adult body in the bone marrow. Those cells can be placed in a medium which facilitates their continued division, so they can live outside of the body. They can divide and form large numbers, and you can then infect those cells with virus, right? And you can get lots of viruses that way, or you can actually study the infected cells to see how they respond to the virus, so you can study cytopathic effects. So cell culture, tissue culture is one way to go there to affect those. An older way that is still tried and true, a classic technique for viral replication, is infecting live bird embryos. So chicken eggs that contain live embryos can be injected with virus. And depending on where you place them in that egg, you can place them directly in the embryo or in the extra embryonic membranes around it. You can kind of tailor that um, infection to some viruses like to develop in the chorion or the amnion or the allantois or some other one of the extra embryonic membranes in that egg. But any place there are cells in that egg, if it's a live egg, can be utilized for viral production. So that's something I think it's a little easier to do than tissue culture. The conditions under which it can be done don't have to be regulated as tightly. All of these, of course, do require very special conditions. They have to be free of contaminants, and so the laboratory protocol has to be pretty stringent to make these work. And then the last thing, which for some people poses ethical problems, is actually using live animals. So sometimes stock animals, sometimes dogs, monkeys. HIV research has gone forward because we can use primate models. We can use rhesus macaques and other kinds of monkeys which are infected by the same retroviruses and study the virus and how it works in those live animals. So I don't know. That's a whole other discussion that goes beyond the boundaries of a science class, but the ethics of that, how you feel about that, may engender some bad feelings. Thinking about people infecting, say, beagles or rabbits or other kinds of animals, but in the name of science, sometimes that is what's done. But that's another way to go. You can infect those animals with virus and study how the virus works. So this petri dish here is filled with these cells that are purple. These are stem cells. They're marked in such a way that you can easily see that they're growing. And they've been infected with virus. And you can see the places here that are kind of cleared out. These are called plaques. And these are sites where the virus has flourished and it's caused lysis of the cells. Okay, so this is what, up here, this is what normally this upper picture B, this is what the cells look like in regions where, they're intact. You can see the individual cells here are nice and visible, and the nuclei are nice and round, and the cells are discrete and separate from one another. So that's what they normally look like. Down here, of course, this is the abnormal situation. This is where there's been a plaque that's formed. So this is a clear area where there are no cells. Here's what's left of those cells. Those are nuclei and other cell parts kind of massed together and form this little piece of debris. So that would be, for this particular virus, maybe something we could look for in an actual human that's been infected with it. So that's something that could be learned by using a tissue culture. This particular virus does that. Other viruses might do something else. They may not cause destruction of cells, they may not make plaques, but they could cause the cells to take on a new appearance or the nuclei to take on a new appearance. So that's tissue culture, right? That's using tissue culture for the purpose of studying virus infection. Here, obviously, this woman is working with live bird eggs inside of a controlled incubation. This is a hood, essentially. You can see there's a little glass barrier, and the air is purified and so forth, so that when she injects those eggs with virus, she's pretty certain that that's all that's getting in there. Bacteria and so forth getting in to kind of contaminate and foul the results of this. And then here, this shows, the potential sites where you can inject viral particles. You got a medium containing viral particles. You can inject directly into the embryo or into the amnion or into other parts that surround it. And that's going to depend on the type of virus because, as I said, they have limited host ranges. And so some of them are real specific for the kind of tissue that they can flourish in. But that's using live embryos for the purpose of viral production. And then the last, well, I said the last slide was the last thing, but apparently there's more. So, viruses obviously, as we're seeing now, have medical importance, and they are the most common cause of acute infections. So when you have an infection and it takes off rapidly and clear it quickly, that's an acute infection. Viruses are the most common cause of those kinds of things. Stomach viruses, cold viruses, those things come and go fairly quickly. And there are several billion of those per year. But then there are some viruses that have particularly high mortality rates. The SARS coronavirus 2 that we're dealing with now seems to have a mortality rate of somewhere between 1.5% to 3%. Depends on the population, and perhaps it depends on the quality of care, what's possible, and depends on the age of the infected person and whether there are comorbidities. There is some evidence that it depends on gender, that males are more likely to be severely impacted by the virus. But anyway, there are viruses that historically have had much higher mortality rates, as high as say 30% or more. But with those viruses, typically the consequences, the symptoms are very serious. They come on very rapidly. So we can see that the person is infected, and we can then isolate them and prevent the spread of it. This particular virus that we're dealing with now causes infections in some folks that are mild. And so they continue to do their normal thing. And while they're doing that, they're shedding virus, and other people are being infected. Or, which is even more insidious, is we have people who are asymptomatic, at least for the first week or so of the period which they're infected. And so again, they can shed virus and spread it to 2, 3, 4 or more people, who then go through the same course of development of the viral infection. And so that's been the problem with this, and that's why we have what we have now, this sort of lockdown and the need to self-quarantine and stay, away from folks and avoid contact with them. So anyway, it doesn't have a real high mortality rate, but we don't have vaccines for it, and this is a novel virus. It's one that none of us have dealt with before. So we have a very large number of people who can be infected by it. All right? And so until it's around for a while and has an opportunity to evolve less virulence, and until a large enough number of us have been infected with it, so that we're now it's going to continue to spread. There are a number of viruses that infect folks that never cause symptoms, right? So that's another interesting thing about viruses. There are some that virtually all of us are infected by. There's one type of virus that you might look up. It's very interesting, called the adenoviruses. Very sorry for my horrible writing with my finger, but adenoviruses. I've learned about these recently. They are viruses that infect humans and, well, other animals as well. And I erased what I'd written up there, didn't I? But they've been discovered in basically all of the major tissues in the body. And they don't appear to cause symptoms. So that's one that may have been around for a long time that evolved toward less and less virulence, and it's the ultimate parasite. It can live in a host and not do harm to the host, and spread and persist without causing problems. Um, there are other viruses that infect us that are associated with things, all kinds of different conditions like arthritis and various autoimmune illnesses. And they can cause chronic afflictions. So sometimes people present with what appear to be, enigmatic symptoms. It's just hard to figure out what's going on with them, and they never do figure out what exactly causes their symptoms. It probably is a virus. It's probably a virus that has never been characterized. So, you have people with weird symptoms that no one can deal with, and it's very frustrating. It's liable to be a virus. Okay, these things are super hard to isolate. So if you have it, it may not have cytopathic effects that are visible. So that's well, that's just in some ways it's amazing that happens, but it does. So what we're saying, we're animals. We're biological entities, and we tend sometimes to forget that, and it seems insulting that we're affected by agents of infection, but it happens. And when population numbers get really large, when host populations get large and dense, these kinds of things can spread very rapidly. And for most non-human populations, when this sort of thing happens, it leads to, unfortunately, decline in population size. It's part of the set of density-dependent factors that regulate population. So we're kind of experiencing that. And this is something that has happened in our past, and it's something that will continue to happen. And it's true that virtually all other organisms have dealt with these sorts of things. So this last statement here is valid, and I think most scientists would agree that viruses are major participants in the Earth's ecosystem. They affect organisms and their survivorship and their fitness, and influence population sizes and population growth rates. And when populations get too big, they're sometimes limited by things like viruses. Anyway, there's that. So that takes us into another area. And I'm liable to put up another video later just about the coronavirus. I'm toying with putting together some specific information about that, not necessarily to test over, but just for you to have, just so you know more about it. Let's move on now. We'll talk about the other types of obligate intracellular parasites, the prions and other infectious particles. Prions are microbial, but they're non-living, as are viruses. But unlike viruses, prions are even simpler. They are simply just misfolded proteins. So normally proteins have a particular conformation, a particular geometry, and that's what enables them to be functional. All right. So if something happens to change that shape, and they become misfolded, it can alter them so that they no longer function as they once did. And they take on new properties that result in the destruction of cells that contain them. And they become extremely resistant to any sort of phenomenon that would tend to break them down. So they resist denaturation. They're impossible really to remove using the typical methods that we implement to disinfect, even going so far as to try to sterilize and use those very, very intense, extreme techniques. Sometimes those don't even work. So high heat, harsh chemicals may not, in case of prions, be efficacious. They may not work. We have to really, really go to the nth degree to take them out. So autoclaving for sure is something we can utilize. There are some very powerful acids and some other oxidizing agents that can destroy them. But outside of that, they're tough to destroy, which is crazy because the proteins from which they form are not like that at all. They're easy to break down. They're easy to denature. So something happens when this change in their conformation, this misfolding happens, just makes them tough. The other crazy thing is they replicate, but they don't use nucleic acid. There's no DNA, there's no RNA. They are strictly protein. And so the parts of the body that they target, for now, it seems to be the brain. So it leads to neurodegeneration. So the brain and central nervous system. So the spinal cord, the brain, those are the parts that they affect. And so diseases that involve the brain and the central nervous system are called encephalopathies, right? The phallo refers to the head. And then pathy, of course, is referring to a disease. So encephalopathies are in the brain. And these are apparently transmissible. So if you have a prion-infected individual and they're shedding, prion, in their saliva or in other body fluids, and those are taken up by another uninfected individual, they too can become infected. And what results then is destruction of brain tissue and other central nervous system tissues, so such that when we look at them under the microscope, they appear to be pocked with little holes all through them. They look like Swiss cheese, whereas previous to that, they were entire, and cells and neuroglia and other things were present throughout that tissue. So when you see big openings, big gaps, big breaks in those tissues, it takes on a Swiss cheese sort of appearance. That's described as being spongiform. So we have these spongiform encephalopathies, and they're just horrible. Now, they lead, in every case, inevitably to death. There's no outcome other than that. They will, in every case, lead to death. They are 100% fatal. How long that takes, varies apparently with the type of prion, but there's no cure, right? And that's, for now, the only way that we know they go is in that direction of death. All right, so some of the prion diseases that have been described. One that goes back a few hundred years is scrapie. That's been around a while, and it occurs in sheep and goats in parts of the world where sheep and goats are commonly reared for wool and milk and meat and whatever else. They've noticed occasionally that their animals, shepherds would notice that their animals would develop unusual behaviors. They would drool. They would ultimately just lose the ability to maintain balance, and they'd fall over and die. So that was referred to as scrapie, and it continues to exist in those animals. And then in cattle, we have the bovine spongiform encephalopathies. There's at least one of those, and possibly others, that infect preferentially the cattle. Bovines are cattle. They would include domestic cows, but also bison, wild forms of bovines. So mad cow disease is the common name for that. BSE is the acronym that's commonly used in discussions and research that look at this particular class of diseases. And then we've got wasting disease, or CWD, chronic wasting disease. It says elk, but it's actually all of the deer. So that family of animals are the cervids. and include our own white-tailed deer that we have here, but mule deer and um, reindeer, red deer, caribou. Of course, reindeer and caribou in the northern hemisphere, especially in the Scandinavian countries, are like cattle are here. They're essential to the life of those folks in those cultures, and they use them for milk and meat and transportation, to pull sleds and things like that. So loss of those animals would be devastating. And moose are also in that family. They're deer and known now to carry CWD. That's spreading. And then a prion that occurs in humans is sometimes called CJS or CJD, Creutzfeldt-Jakob disease, is a prion type of infection that occurs in humans. And there are others that affect humans preferentially. One called Kuru, which appears and has appeared, I don't know how frequently it appears now, but it has appeared in cultures, tribal or aboriginal cultures, and say historically practiced cannibalism. They don't do that so much anymore, but if you were to do that and eat the brain of, say, one of your enemies after a battle, you might acquire that, and that was noted in some of those populations. So there's that. But this shows the neuron soma, a nerve cell, shows a cell body, and shows the normal proteins that are soluble in that cytoplasm. When the prion enters, the prion's got a different form. When it enters, it literally causes these normal proteins to change form as well. There's some way that they interact so that the prion proteins stimulate the development of more prion protein from normal proteins that are there. So ultimately, the normal ones become less and less abundant. It sort of trends toward more and more of the prion. The prion numbers build up, and they start to accumulate along the margins of the cell. Now, if you know anything about how nerve cells work, that cell membrane is where action potentials arise. You have fluxes of sodium and potassium across that membrane, which that's the basis for nerve transmission. So if you have a bunch of prions laying in there, you can't develop an action potential. There's no way that nerve signals can pass down that axon. So that neuron and others around it likely are going to be shut down and lead then to paralysis and ultimately death of that individual. All right, so there are those. And then we've got these other types of infectious agents, some of which don't really even cause symptoms, but we see them showing up in cells and multiplying. So satellite viruses are short little pieces of genetic material. Got adeno-associated viruses. We have this whole class of adenoviruses which cause symptoms in the upper respiratory tract, similar to what we see in the common cold and other somewhat more severe viruses. But always when those show up, we see these other small pieces of genetic material that appear to be adeno-associated viruses. And then there's delta agent, little pieces of RNA that are expressed in the presence of hepatitis B virus. They're not the hepatitis B virus. They show up together with it typically. Those are called satellite viruses. So the key is another virus has to be present, infecting a cell before these satellite viruses can themselves reproduce. It kind of sets the stage for their reproduction. And then a viroid is a really short piece of RNA that doesn't have a capsid that apparently can replicate and be passed from host to host. So those are viroids. And they can cause some symptoms in plants, but none that are terribly devastating. In fact, it just simply alters the appearance of plants. Sometimes the leaves that are normally solid green will take on a mottled appearance, the presence of these. So, if you've got a leaf with little gold spots or flecks throughout it, it may indicate that those cells have been infected with viroids. And those are only known in plants. All right, so there's that. That's where I'm stopping this. So that's the last lecture. I'll post more information about when an exam is going to come up on this, but the next exam will be just over the viruses, right? The next unit that we'll get into looks at ecology and the conditions that viruses need to persist. So I'll be going over that as well. But that'll be the next unit for the next exam. This next one, exam 3, is just going to be over the viruses. So I'll be letting you know about when to expect that. If you do it, you're going to have a week to do it. I'm going to try to make it as accessible and painless as possible. All right, so I've got to let my dog in. I'll talk to you later.